This is Dr. Mobeen Sayed. Today we are going to talk about MHC I loading. So it is done for the endogenous antigens that include self-antigen and cancerous antigens and any infective pathogen antigens. So let's talk about that. So major histocompatibility complex 1. The primary objective of the discussion today is how it is loaded and presented onto the surface with an antigen. So in that process, we'll discuss how it is manufactured as well, plus we'll look at its properties too. So if you look at the center part of the diagram, it has an alpha chain, which contains alpha 1 part, alpha 2 part, alpha 3, and a membrane anchoring part. There is a separate protein called beta 2 microglobulin that is also attached to this with non-covalent bonding. Beta 2 microglobulin actually helps to stabilize the remaining complex. The MHC Class I complex binds with CD8 T cells or cytotoxic T cell. Remember that MHC number, for example here Class I, multiplied with the T cell should be 8. So here the number is 1, so the T cell has to be 8. If the number was 2, for example MHC Class II, then the CD4 cells will bind with that. Now MHC Class I is presented on all nucleated cells with certain exceptions. Of course, it is not present on anucleated cells, for example RBC. However, sperms also do not express this, or a study showed that out of all the sperms, 24% may express this. Some neurons do not express this or express it in reduced amount. Some immune privileged sites, for example retina, may not express this, or if it expresses MHC I, it tries to express them less, so there is not a lot of inflammation which could then cause retinal damage. Also remember that as much as we say that antigen presenting cells, the professional antigen presenting cells, that is the dendritic cells, macrophages, and B cells, they present antigens on MHC II, the exogenous antigen, the antigens that are present outside of the cell. We discussed that with the MHC II loading. However, please remember, sometimes cells like dendritic cells can actually take an exogenous antigen and cross-present that on MHC I as well without being infected. This is called cross-presentation. The benefit of doing this is that dendritic cells can now activate not only the humoral arm, the CD4 arm, and the B cell arm, but rather cytotoxic arm as well. So they can present the exogenous antigen on MHC II and MHC I without being infected. So this is called cross-presentation. Okay, so the genes. As we discussed in MHC II as well, on the chromosome number 6, small arm or petite arm, we have class 1, then class 3, and then class 2 genes. These are also called HLA Class I, HLA Class III, and HLA Class II. The class 3 genes are actually between the class 1 and 2. Class 3 genes, I've not made them here. They are usually busy or involved in cytokine production, complement production, etc. Here we are talking about MHC I. The genes for the MHC I are B, C, and A genes. They all make alpha chains. They do not make beta globulin. Beta 2 globulin is made by the genes that are present on chromosome number 15. So also, as I said last time too, in MHC II class loading, if the gene is one letter word or named ABC, that is class 1 MHC. If it is two letter DP, DQ, DR, then that is class 2 MHC. Just to remember that for your exams. Now, for the loading process, first let's look at the cell. So here is a nucleus. Next to the nucleus is the rough endoplasmic reticulum. Rough endoplasmic reticulum means that we have the reticulum here, which is studded by ribosomes, which will be helping with the protein translation. Now remember, the ribosomes can be free floating in the cytoplasm as well, and the messenger can come out, messenger RNA can come out in the cytoplasm as well. So there can be proteins that are manufactured in the cytoplasm. However, those proteins that need to be secreted are usually manufactured within the lumen of the rough endoplasmic reticulum. In the case of the MHC I and the antigen that is loaded on it, there is an interesting problem. The problem is the antigens will be found in the cytoplasm, while MHC I will be created in the rough endoplasmic reticulum because it needs to be expressed on the cell surface. So how will we bring the antigen from the cytoplasm to the MHC I, which is in the rough endoplasmic reticulum? We'll discuss that in a second. And then here is the Golgi apparatus. It is necessary for anything that will be secreted from the cell or expressed on the cell surface will have to be processed by the Golgi apparatus. And I made a little thing over here called proteosome. You know that proteosomes are little enzymes that are present and they will break down proteins. So that is why they're called proteosomes. So with this structure in mind, let's now see how loading occurs. So maybe you can take a screenshot of this. So imagine that this is a cell which is doing its regular business, and that maybe it is making its own normal proteins, or it is infected and there are some viruses, copies that are present in the cytoplasm, or it may be cancerous and it is developing cancer proteins. So let's see how this whole process works. So don't be too scared of this diagram. It's actually very easy. Here is the nucleus. Nucleus has nucleopores in it. These are tiny holes from which messenger RNA comes out. Those proteins, so here, this is the rough endoplasmic reticulum. This reticulum has ribosomes attached with it. That is why it looks rough. There is smooth endoplasmic reticulum as well. We're not talking about that at this time. This is the Golgi apparatus. This is the cell membrane. Now, what happens is those proteins, for example, MHC I protein, needs to be expressed on the cell surface. So the messenger RNA to make MHC I protein will come out of the nuclear pore and directly enter the rough endoplasmic reticulum. From there, it will enter a ribosome, or it will become attached with the ribosome, and then the ribosome will help make MHC I proteins. We'll talk about that in a second. Let's first create an antigen. So imagine that this is the messenger RNA that came out of a cell to make a cell's protein, and that messenger RNA will bind to a free-floating cytoplasmic ribosome, and there will be some proteins that will be formed within the cytoplasm. Similarly, imagine that we have a virus, let's say an RNA virus. RNA virus will release their RNA within the cytoplasm of the cell, and those RNAs will bind with our ribosome and start making viral proteins. DNA viruses can actually create messenger RNA from within the nucleus as well, which would then come out and bind with the ribosomes. So here, what is important to notice is that, let's say it's a virally infected cell. So here is the viral RNA, got bound with the ribosome. Ribosome did the translation. This is happening with the free-floating ribosomes in the cytoplasm. And the viral protein is formed. Many virus copies are assembled. However, some of those proteins will actually be picked up by proteosomes. Proteosomes will randomly pick up anything that is in their environment. It doesn't matter it is a viral or bacterial or fungal protein. The proteosomes can actually pick up our normal, regular proteins as well and help present them on the cell surface as well. These are called the self-antigen. So here is a proteosome. Proteosome will pick up the viral protein. We're talking about viruses here, or bacteria, fungus, and then it will shred that protein into pieces. It would break that protein into smaller pieces, and those pieces now need to get into the rough endoplasmic reticulum, or just simply endoplasmic reticulum, to be loaded on the MHC. So how are these proteins brought in here? So if you see here this little transporter that is standing here, this transporter is called Transporter Associated with Antigen Processing, or TAP. Transporter associated with antigen processing or TAP. This transporter will pick up these antigens. Once again, could be self-antigen, viral antigen, bacterial antigen, fungal antigen, cancerous antigens. And these transporters will bring these antigen, and they are given a way to get into the rough endoplasmic reticulum and unload those antigens here. Now, let's forget about this part for a second, and let's now build MHC I. For MHC I, what will happen is that the messenger RNA for the MHC I will be released from the nucleus into the rough endoplasmic reticulum. Over there, the messenger RNA will get loaded onto the ribosome. Ribosome will then do the translation and make the MHC I proteins. What are those proteins? Really, the alpha chain. Alpha chain has three pieces: alpha 1, 2, and 3. So if you see here, this is the alpha 3, this is alpha 2, and this is alpha 1. When they are created, these complexes, they are very loosely bound with each other and they are shaky. They are not able to really bind with an antigen because they're loosely attached. So they need cooperation from the beta 2 globulin to come in and get attached to them and provide them a structure which will then become stable and solid. So not solid like metals, protein solid. So beta 2 globulin, that messenger RNA is also coming in from the nucleus. It was developed from the gene on the chromosome 15. That would come in, and the beta 2 globulin will be formed in the rough endoplasmic reticulum. All of this will then complex together, making the MHC Class I complex. Now, there are special loader proteins. Those loader proteins will, just like, if you are trying to load a truck or something, and there may be many people who are helping you put sofas and couches on the truck. So same way over here, there are loader proteins that will pick up the antigen from the TAP, the transporter, and help load them onto the binding site of the MHC I. This binding site is interesting. It is more restricted compared to MHC II. MHC II can actually bind larger peptides. However, MHC I binds about 8 to 10 peptide long proteins. Interestingly, one binding region of MHC I can actually bind multiple kinds of antigen because the binding region is not very restricted to full shape. Instead, if this was the binding region, it will have some anchor points on it where the amino acids of the antigen will attach, but the remaining can be loose. So an antigen that may be shaped like this can attach here. An antigen that can be shaped like this can attach here. An antigen that can be shaped like this can attach here. So multiple kinds of antigen can be attached to one binding site. This is the same case for MHC II as well. MHC II is even more flexible, that it can actually allow longer peptides to be attached. Okay, so now we've gotten the MHC I, we've gotten it stabilized with beta 2 globulin, then we have gotten the help of the loaders to load the antigen onto the binding site. Then what will happen is that from the rough endoplasmic reticulum, small secretory vesicles will be formed, which will then move the vesicle from the rough endoplasmic reticulum towards the cis phase of the Golgi apparatus. So now that vesicle would end up in the Golgi apparatus. Once inside the Golgi apparatus, it would be further processed and then released outside from the Golgi apparatus. Now it will be marked to be brought to the cell surface. So once it is marked that way, that vesicle would come in. So let's say this was the vesicle. It will come in and will fuse with the cell membrane. The membrane anchoring part was already attached to the vesicle membrane. And so that is the same attachment over here. And now the MHC I will be expressed outside with the antigen loaded on it. The cytotoxic T cells would then come and attach with MHC I, and they would attach or bind with the antigen. So that is the process of loading of the MHC one. The same is the process. I used viruses as an example. The same is process for the fungus or bacteria or self-antigens or even the cancerous antigens. Now, there are some interesting things. Imagine that we have an antigen that has been brought in by the transporter, but the antigen is bigger in size and it will not fit on MHC I. In that case, the antigen is thrown out or ejected from rough endoplasmic reticulum into the cytoplasm, where either it will need to be trimmed to size, meaning it would go back to the proteosome, will get shredded further and then brought back in, or it will be ubiquitinated and destroyed. Similarly, there may be antigens that are brought in here in the rough endoplasmic reticulum which cannot bind with the MHC I because they just do not have the interactive units that can bind with the anchor points on the MHC I. In this case as well, those remaining proteins will also be removed and they would be ejected to cytoplasm for ubiquitination and destruction. So that means not everything that is present in the rough endoplasmic reticulum will become presented on the MHC I, only those that can bind. And there would be a chance to trim them to size and bind them, and if that doesn't happen, they are just simply destroyed within the cell. Now, some notes to keep in mind. Some viruses play tricks with this MHC I to try to evade immune responses. For example, HIV virus can actually internalize or endocytose the NEF protein by the HIV virus, can trigger the endocytosis of the MHC that are already expressed on the surface. And so what it does is it is afraid, the HIV virus, that the immune system cell would now start finding that the virus is in here and kill this cell. So instead of doing that, what they do is they simply trigger the endocytosis of the MHC I, so the cell is presenting less MHC I, and hopefully that way HIV tries to evade, and it successfully does that. Similarly, some adenoviruses, they bind to MHC in the endoplasmic reticulum and they prevent its exit. So they bind here with the MHC within the endoplasmic reticulum and they do not let it be secreted all the way out. So that is how they try to evade detection. Similarly, human cytomegalovirus, that can eject MHC I to cytoplasm for degradation. So remember that I said that some proteins that cannot be loaded on the MHC I will be kicked out. This virus can actually cause the MHC I to be kicked out. And once the MHC I is out in the cytoplasm, it is in the wild, wild west. It will actually be degraded and killed. The human herpesvirus 7 binds to MHC I in the rough endoplasmic reticulum and routes it for lysosomal degradation. Similarly, Kaposi sarcoma-associated herpesvirus ubiquitinates MHC I, which targets it for destruction as well. SARS-CoV-2 can reduce the induction of MHC I pathway. So there are various ways the viruses play tricks on MHC I to try to avoid detection. So this is the discussion about MHC I and how it is loaded and how it presents this antigen on the cell surface. Thank you very much. I'll talk to you later.